A very warm good afternoon to everyone joining us on this very special Sunday afternoon as we are here to talk on a very important topic with one of the pioneers in the field. So what is today's talk all about? It is about COVID-19 in India, mutations, genomics and reinfections. It's our pleasure from TREAT by ALTERDOCTORS to welcome Dr. Vinod Scaria. He's the principal scientist at the CSIR Institute of Genomics and Integrative Biology to help us address these issues today. He's really a pioneer in precision medicine and clinical genomics in India. He has been one of the first to sequence the entire Indian human genome along with his collaborator, Dr. Sridhar. Now, Dr. Scaria has been pioneering the application of genomics to diagnose also and set up really a group to look at rare genetic diseases in India. Now, today's talk is all about COVID-19, and really, he has been pioneering the effort with the COVID-19 Open Research Data and Resource, which is an initiative basically to make relevant data, research, and resources to be available in an open format to ensure not only that it is widely accessible, but also it is accessible in a timely way. So without much further ado, I think we shall move to the topic of the day for which people are excitedly waiting. Dr. Scaria, I thank you again so much for agreeing to do this. The virtual stage is all yours.
Thank you, Nitin. So over the next 15 minutes or so, I'll try to give you a basic glimpse of what is genomics of SARS-CoV-2 all about, and some examples, not all examples, some examples where you can actually make sense out of these genomic data. So to start, it is really important to acknowledge that we stand on the shoulders of other researchers who made the basis of all of this. Thanks to researchers from China who made the sequences of SARS-CoV-2 available very early in the epidemic. And this is the basis of practically all the genomics that we do and have been done across the world. Now, what's really important to note in this particular episode of this particular pandemic of SARS-CoV-2 is that typically when we, as clinicians, talk about microbiology, we always talk about isolating an organism and then doing a particular test on an isolated organism, whether it is a bacteria which is cultured, or for a virus which is cultured in a cell line, or so on and so forth, which typically is a very tedious, time-consuming, and also human-intensive activity. Now, what really changed in between in the last half a decade or so has been the throughput or the availability of genomic sequencing that made this entire problem turn on its own. So if you look at the paper which described the first SARS-CoV-2 virus genome, what they did was to essentially take samples from the bronchoalveolar lavage, which is essentially the raw sample. You could directly do a total RNA extraction, ribosomal RNA depletion, because the ribosomal RNA from the humans are quite large in number. And you could directly go back to do sequencing. And then, of course, the rest of the bioinformatics, which includes assembly, analysis, and of course, the phylogenetic analysis. Now, what I'm trying to say here is that you don't really require to do the sample isolation, the sample enrichment by culture that we typically used to do in microbiology anymore, thanks to the advent of genomics. Now, the other concept that I need to bring to you is that the virus is constantly mutating, and like any other organisms, whether it is human or it is a bacteria or it's a virus, we are all generating or we are all evolving by accumulation of mutations. The only difference is the rate at which we accumulate mutations. Viruses, of course, accumulate more number of mutations at a faster rate than humans, and then therefore they have a chance to evolve much faster. To just give you an example, the first Wuhan Q1 isolate was collected from December 26th. By Jan 8, you would see isolates would have accumulated an additional mutation. And for example, in February 27, there are multiple mutations that have been accumulated in the same virus. So there's nothing to be alarming about it because viruses do mutate. The rate of the mutation is pretty much constant. The virus mutates approximately at the rate of one mutation or one substitution every week or so. Now, this is the basis of all what we do in genomics, that there are just a few genetic mutations which are different and they originate at a definitive sort of rate on the viral genome, and the viral genome is quite large, 30,000 base pairs. So these are not really going to change SARS-CoV-2 to something else, but essentially slowly accumulating mutations which can confer them additional functional properties. Now, the other thing that you need to keep in mind is that when the virus mutates or accumulates substitutions, and this is at a constant rate as said before, uh, the virus could accumulate mutations as they move on and diverge out. So for example, if we keep the mutations as positions of 1, 2, 3, 4, 5, and 6, as the virus gets transmitted from individuals to individuals, they keep accumulating mutations. And for example, an individual transmits to two individuals, they diverge at that point in time, and then therefore they become their own lineages. Now, essentially in genomics, what you're going to do is to take a snapshot in time and then look at the relative proportions of each of these groups, or what we call as haplotypes. So when I say 1, 2, 3, 5, that essentially means that it can trace a lineage from 1, then we know that 2 was in between, then we know there was a divergence at the point, it became 3, and then it accumulated 5. And there was another branch point at that point in time, which was 1, 2, 4, and 6. So looking at the viral genome, you can sort of trace the lineage or the ancestry of the virus. And this is what we call as haplotypes. Now, typically, if the virus was emerging at a constant rate or was transmitting at a constant rate, you would have equal proportions of each of these haplotypes at any point of time. So a point in time is something that you can look at in a vertical space. So if it was a normal kind of a distribution, you would have a distribution of these haplotypes which are normally distributed across any point of time. Now, there are some points in time where the virus emerges or that makes a very large outbreaks. In other words, these are essentially also called super spreader events, where a particular haplotype of a virus emerges into a very large proportion. And that's how we can actually go back and look at outbreaks or locations of outbreaks where, where you can go back and intervene. So this is another principle that you need to keep in mind. Now, practically all of genomics that we do is based on these two principles. Now, if you look at all the viruses out there in India, there are today around 3,600 odd virus genomes sequenced from India, thanks to multiple different labs of the CSIR system, the DBT system, and many other laboratories from the states which made all this data available. Now, globally, there are 10 clades or lineages of the virus, out of which 6 of the clades or lineages are also found in India. Apart from that, India also has a unique or a distinct lineage, which we call as the I or the A3I. Now, I or the A3I as a large distinct lineage is not found elsewhere in the world, though there are genetic sequences which belong to this particular lineage in very small numbers in the rest of the world. And this rest of the world could include regions like Southeast Asia, mostly Southeast Asia and Australia, and of course, in some cases from the Middle East. Now, since this is a very distinct and large cluster or clade or the lineage of genomes from India, we sort of named it as I, signifying the country, and you also named it as A3I because it seems to have diverged out of the A3 lineage of the virus. So what you need to keep in mind is that apart from all the other 6 lineages, India has a distinct lineage which is called the IA3I. Now, the relative proportions of virus of these lineages are also distinctly different in different parts of the country. Now, the largest proportion of isolates belong to the A2A clade of the virus, which essentially is a European lineage of the virus. And today, around 70% of the virus lineages are from the A2A. Now, depending upon which state you look at, the relative proportions of A2A and IA3I are distinctly different. For example, if you look at Delhi, the majority of the isolates belong to the IA3I, and the next largest proportion is probably from Telangana. But if you look at, for example, Gujarat or Maharashtra, the largest proportions of the virus belong to the A2A clade. And what we also know from the divergence estimates is that the IA3I essentially came out from an outbreak. That's the right-hand panel of the slide, and this outbreak spread to practically a large number of states across the country, and then therefore they diverged out. Now, of course, you can build haplotype maps. So I told what a haplotype is. You can build haplotype maps of genomes across the country, and this is how it would look like. Now, what you need to keep in mind in this haplotype maps is that you can now look at how did a particular epidemic emerge and contribute to the sample numbers in a particular state at a particular point in time. So each of these large dots out there are essentially points of outbreak. So you could see an outbreak, for example, in Kerala. You could see an outbreak, for example, in Telangana. You could see an outbreak, for example, in Karnataka, so on and so forth, and Orissa, and so forth. And the colors essentially represent which are the states which are involved in that particular outbreak. So what you realize here is that many of the viruses that are circulating today in the country are actually a result of local outbreaks in contrast to people who have brought it internationally. Now, frankly speaking, all the virus that has come into India has come in through people who have traveled from outside the country, but that necessarily doesn't mean that all the proportion of or all the viruses are essentially contributed by them. At some point of time, some of these individuals or the secondary or tertiary contacts of these individuals would have contributed to an outbreak or an emergence event. And from this emergence, a large number of individuals get infected, and then therefore they spread the virus in the local population. Now, so looking at the haplotype maps, you can now understand where did this emergence happen and how did it contribute to the total number of cases in a particular state or in a particular district. So that is what we call as molecular contact tracing. In other words, if you look at a real-time PCR, you can only say whether it's a positive or a negative. It cannot tell what is the background from which this individual came. So if you know that there was an outbreak event, you can now quantify what does that outbreak contribute to the total number of cases in a particular state or a particular district. So that's what we do in terms of molecular contact tracing. Now, all of this data is available in a browsable format. This is Indico, which is a resource that we maintain. And you can go back and look at this resource and sort of parse through the genetic variants. It provides a lot of information about the genomes from India and the genetic variations out there. And the other thing that you need to think about is also how do we really build an application of scale comparable to the applications that you do, for example, real-time PCR, to be able to build genetic epidemiology of the virus. And this is essentially work of my colleague, Dr. Sridhar Sivasubbu, who is also my close colleague and collaborator at IGIB. And the idea of building this application was how do we do sequencing of a very large scale so that we can enable genetic epidemiology at every state, or probably at every district, at a quite affordable rate. And this is an assay that we sort of clinically validated for the first time, probably also in the world. And the idea of doing this was to make what you call a seamless process where you could do genetic epidemiology. It starts with a tube which has a barcode and an app where you can sort of give your consent and enter the barcode. You can do a self-sampling. You don't need clinicians to do sampling. And we have done this over thousands of times now to realize that people can do sampling on their own. You don't really require a clinician to sample an individual. You can just do self-sampling, put it into a tube, and send it over to the lab. And the lab, what we can do is to sequence these viruses at a very high throughput. And today we can sequence over 3,000 viruses in around 12 hours time, and at a cost which is comparable to real-time PCR. And of course, there's an added advantage. It's much more sensitive and specific than real-time PCR. And the lab, of course, it goes through a much complicated step. Some part of this are automated. So they're essentially going into library preps, and then the sequences get generated. The sequences are analyzed by a sort of automated computational pipeline, some of which are enterprise and some of which are built in the lab. And at the end of the day, you can get a phylogenetic analysis to understand which cluster or what outbreak has contributed to this case. And of course, you could also generate a report which can go back to the patient and which is displayed on the app. Um, and of course, there are a variety of things addition to all this, what we can do. And what's the most important and pertinent of the many applications that we have sort of tried, and I think that all of you should know, is that all of what we do in COVID testing is based on a concept called RT-PCR. And RT-PCR essentially means that you have primers or probes which can specifically bind to a region in the viral genome and amplify a stretch of the genome, and you essentially probe these stretches which are amplified to understand whether the virus was there or the virus is not there in a particular sample. Now, all of this is dependent upon the fact that you have primers of the probes which bind to the viral genome, and the binding to the viral genome could also be influenced by genetic variations. So what we essentially did was to go back and look at all the primer and the probe binding sites in the viral genome and ask a very simple question: are there genetic mutations which can change the efficacy of this binding, and then therefore could contribute to false negative results? And to surprise, what we found is a significant number of genetic variations do fall into the primer and probe sites, especially the ones which are used in India. And then therefore, we gave out this dossier to some state governments which are interested in looking at these viral profiles, at least in the southern states of Kerala and Tamil Nadu. These have actually gone back to the governments, which now goes back to the labs to decide what are the reagents that they should procure, or what are the primer or probes that they should not use in the lab, because a significant amount of the viruses from that particular region would have genetic variations out there. So this is one application where, and an immediate application where you can really use genomic sequences to create evidence, and based on the evidence, write policy on what the tests that you should do and what are the test kits that you should not be using in your clinics. I think I'll stop there. And before I stop, let me acknowledge a lot of people who made this possible, a lot of people who have worked from home during this tough period, and a subset of people who stayed back in the lab despite the risk of working with coronavirus. And of course, this is all possible with generous funding from CSIR and fantastic set of clinical collaborators and research collaborators in IJB and across the CSIR system.